That's a lot of people missing, eh?
How many are there? 4?
Only like 18.
On the thing? Yeah.
Do you think this one? Okay. We are going to start chapter 2 today. And so we're going to start looking at energy systems and energy substrates, and which system and which substrate is predominant at what time during moving and exercise and physical activity. Okay? I have to be honest and say that I have not had chance to look at this chapter in the new book. So I'm really hoping, I'm using my notes from the previous edition, I'm really hoping that nothing significant has changed. If you've read the chapter and I say something that you think is different from what you read in the chapter, please let me know. That would be great. And I'll catch it up for next week. Okay, let's have a look. Okay, so chapter 2 is looking at anaerobic systems. So these are energy systems that do not need oxygen in order to produce the energy for muscle contraction. And then chapter 3 looks at the aerobic system of production using oxygen. So our learning objectives for the chapter. And so we use plants and animal products for fuel. The fancy term for turning food into a form of energy is bioenergetics. You can also use the term metabolism. They're interchangeable. Okay? And hopefully you remember from school that ultimately we're reliant on the sun, and the plants convert that sunlight into carbohydrate, and then animals eat the plants, and it all recycles very nicely. So we've got chemical, electrical, heat, and mechanical types of energy. When we're looking at XPhys, we're going to be looking at mostly the first 2, but we talk a lot about the production of heat during exercise. All right. So, our energy substrates, right? What do we turn into energy for muscle contraction? So the first one we're going to look at is carbohydrates. And carbohydrates are great for immediate sources of energy. They're very easy to break down. They're relatively small molecules. They're easy to store. And so, that's our go-to choice for energy in most situations. So we can have either monosaccharides, disaccharides, or polysaccharides. The polysaccharides are the starchy cellulose ones, and also the glycogen, which is the storage form of carbohydrate in humans. All right? So glucose, which is a monosaccharide, is the most important carbohydrate from a metabolic standpoint. Monosaccharides are called simple sugars. There's not much to them. They break down very easily and release their energy bonds. Okay? We want to think, no, let me come back to that. I'll come back to that idea. Glycogenesis, glycogenolysis are fancy terms for making and breaking down glycogen, all right? That can occur either in the liver or in muscle tissue, which are the two places where glycogen is stored in the body. Okay, here we go. So, we've already mentioned glucose is a simple sugar. Up here on the top right is a diagram of a glucose molecule. Now, this is not a chemistry or a biology class, okay? So you don't need to be able to draw that. You don't even have to know the form of it for this class, unless that helps you, right? If you're the kind of person where the tiny details are really beneficial to your learning curve, go ahead and use it. What I'm interested in is the fact that we have a 6-carbon ring, all right? So there are 6 carbons in a glucose molecule, and that's important later on when we look at where do we produce the most amount of energy from, all right? Glycogen, as I said, is the storage form of carbohydrate, and that's stored mostly in the muscle tissue and in the liver. And glucose and glycogen are really important for metabolism at rest, but also during exercise. So we'll come back to that idea. The glycogen in the muscles provides the substrate for energy production within the muscle tissue. The glycogen in the liver gets broken down into glucose to restore blood glucose levels. So if I'm doing some form of exercise where I'm using a lot of carbohydrate, then the liver will break down some additional carbohydrate and top up the blood sugar levels to keep the brain happy and to keep feeding your muscle tissue. This molecule down here is a glycogen molecule, and each one of these little red hexagon, 1, 2, 3, 4, 5, yeah, hexagons is glucose, right? So the glycogen molecule is just made up of glucose and glucose all stuck together. Our second energy substrate that we can break down and access energy bonds from are fats or lipids, right? This diagram is an 18-carbon fatty acid. So you can see, again, it's not important. You don't have to be able to draw it or any of that. What's important is look how many carbons there are there compared to our glucose molecule, all right? So we can metabolize fat and use it for energy under certain circumstances, and we can find fats in both plant and animal tissues, different types of fats, right? So the storage form of carbohydrate was glycogen. The storage form of fats is triglycerides, okay? And when we look at fats, they're going to have an even number of these carbon atoms that I'm interested in, okay? And they can be saturated, mono-unsaturated, or polyunsaturated. Those are our 3 types of fats. Right? When we store them, they're stored in fat cells, in adipocytes, also in the muscle tissue, right? And a triglyceride is a glycerol plus 3 fatty acids. So when we want to get at the energy, we have to break that triglyceride down to get at the energy bonds in the fatty acid. Right. Monounsaturated and polyunsaturated are preferred typically from a health benefit standpoint. Most of the recommendations, if you look at any of the national governmental websites like the Center for Disease Control or the American Heart Association or the NIH, the recommendations are not to cut out saturated fat altogether, but to minimize how much of it you have in comparison to the other two types. All right, so we want to prioritize monounsaturated and polyunsaturated fats in our diet typically. Right? So even in someone who is very lean, fat stores are abundant. We have a lot of fat stored away, and so we can't ever really deplete it. This is a different picture from our carbohydrate. Our carbohydrate is actually a very finite store of energy, right? It's like a pantry, and once I've used it all, I have to replenish it on a regular basis. So we have to constantly replenish our stored carbohydrate. We don't have to do that with fats because we've got plenty of them, okay? So in this little table here gives you a good idea of the difference between how much energy is stored as carbohydrate and how much energy is stored as fat. And I think it's important to note that this is based on an individual who only has 12% body fat, so probably a male, because as we saw in chapter 13, 12% body fat is very low for a female. So this is probably a man, all right? But still, 12% body fat is pretty low, okay? And I think that's important when we look at the amount of energy that's available, okay? So in the liver, we have about 450 kilocalories of energy stored. In the muscles around our body, we have just over 2,000 kilocalories stored, and then there's somewhere between 50 and 100 calories stored in your bloodstream as blood sugar, blood glucose, okay? So somewhere in the region of 2,500 kilocalories at any one time if your glycogen stores are topped up, okay? Now, again, thinking about this person being quite lean, all right? We have nearly 73,500 kilocalories stored in fat cells under our skin and around our trunk. And about 1,500 kilocalories stored within the muscle tissue. So that's about almost 75,000, right? 73.5, 74, 75, and only 2,500 for carbohydrate, right? So you can see that fat has lots of energy. The difficulty with fat and using it as an energy source is that the molecules are very, very large. So it takes a long time to break them down to the point where we can access the energy bond, right? That's why carbohydrates are so useful, because they're not that large. It's very easy to get at the energy bonds, right? They break down very quickly. Okay, so energy substrate number 3 is a little bit controversial in that right now it's very popular as a diet And there's lots of nonsense around about what we do and do not use proteins for. Okay, so proteins we can find in animal products. We can also find proteins in plants, although there aren't a lot so far that we know of that are complete proteins. So like edamame or soy, tofu, that's a complete protein. So it's the equivalent of an animal protein. But most plant proteins are bits of proteins, and we have to be careful to select the right ones and put them together. Right? So it's a problem if you're a vegetarian. You have to be very clever with your combinations of your foods. So there's somewhere around 20 amino acids that are kind of Legoed together to make different proteins. Okay? Proteins act like a building block rather than an energy source. Right? We use proteins to fix things when they're broken. We use proteins to grow things if we're trying to get larger muscles. We use proteins to make hormones so that our endocrine system functions properly. Okay. Of our amino acids, 9 of them are called essential. And they're essential because we can't make them in our body. We don't have the ability to synthesize any of that group of 9. So that means we have to make sure to include them in our nutritional intake. Right? The remaining 11 or so are non-essential. So that means if they're not in my food source, then I can synthesize them from other things within my body. Right? So we have to be careful, particularly with the essential ones, because without a particular amino acid, the protein that needs it to build the protein can't work properly, right? So the protein can't do its job because it's missing, bless you, missing an amino acid, all right? So it's important to identify that you're completely covering at least the 9 essential ones, okay? So what's crucial here is this last statement, because it's, as I said, right now there's a lot of nonsense around about using protein for energy. We do not use protein for energy as human beings. Very rarely, right? We use carbohydrates and we use fats, right? Preferentially. So we can metabolize protein for energy, but we typically don't metabolize protein for energy. We use it for all these other jobs, all right? So let me give you an example. If you run a marathon, okay, so let's say that's 26.2 miles, say it takes you 3.5 hours to run a marathon. About 5%, somewhere between 5 and 10% of the energy you need for 3.5 hours of running might come from protein. Right? If you go down to the max pack and you work out for an hour, you don't use protein for energy. Okay? So keep that in mind, because as I said, there's a lot of marketing nonsense around right now about protein and energy. All right, the other molecule, I guess, that we want to understand are enzymes. Now, enzymes are not an energy source, right? They're not a substrate. But without enzymes, we wouldn't be able to break our food down quickly enough to get at that energy as we need it. So enzymes, when you see a word that has A's, ASE on the end of it, that's typically an enzyme, right? So that ASE identifies enzyme. And enzymes are proteins, so here's a good use for our protein, right? And they don't cause metabolism, but they facilitate metabolism. So when the enzymes are available in the environment, then the metabolic pathways work more effectively, okay? And they use less energy to create energy. Right? So typically, they're what we call a lock and key mechanism. So an enzyme is very specific to a job, right? They don't kind of job share. One enzyme has one job to do, right? And so there are lots of enzymes in our body. They can be responsible for facilitating catabolic reactions, so breaking down carbohydrate and fats to get at proteins, or anabolic reactions, forming products, so making glycogen from glucose. Ooh, let me go back. Any questions so far? Nope. Okay, thank you. All right, so another interesting thing about enzymes is not only are they very job specific, they're really fussy, right? They're very temperamental. So they work best at a certain temperature and a certain level of acidity within the cell. So if we increase temperature very slightly, then it increases the speed and the energy production because it increases the enzymatic action on the substrate, right? If we increase acidity, then they don't like that at all, right? They're very, very unhappy if acid levels go up or pH drops. So that would decrease their activity levels and therefore decrease energy production. Now this idea becomes really important when we get to the second energy system, all right? Because if I've got decreased energy production, then I'm looking at physiological fatigue, okay?
Ahmed raised his hand.
Who did?
Ibn.
Yes, Ibn. Did you have a question? Is he typing anything?
No.
No?
He's reconnecting.
He took it down?
He's reconnecting, I think.
Oh, okay. Is he back in? Uh-oh. Okay, I'll keep going and maybe when Ibn reconnects, he can ask his question.
I'm back.
Oh, he's back. Hi there. What was your question?
So I was curious to, is the enzymes that are in our bodies that break down the foods, are they the same as the ones that we intake from other foods? Or like, what is the difference?
Some of the enzymes that we need in our bodies we would be making from protein. I'm not sure if we digest enzymes that we eat from other... I don't know the whole answer to that, Ibn. I would have to do some research. We definitely make some enzymes using proteins. right? So that's one of the instances where if there was an amino acid missing, then that protein wouldn't be able to play a role in making the enzyme that we need. But do we digest enzymes that we eat? We don't know. That's outside my sphere of knowledge. I will have to look that up.
Okay.
So I will see if I can get an answer for you on that. Good question. All right, so we've been talking about energy, right? What is it precisely that we're talking about when we look at the human body, okay? So the energy that we can utilize for muscle contraction is called ATP, adenosine triphosphate, okay? And we have other energy molecules available in the muscle cells, but the only one that we have evolved to use is ATP, okay? So that makes it the most important one for us, and it's what we're looking to make when we're looking at these energy systems, all right? So when we're looking at our anaerobic energy systems, we want to know how do we make ATP for muscle contraction when we're not using oxygen, and then in chapter 3 we'll look at how do we make ATP when oxygen is available. All right? So ATP, again, you don't need to know this diagram of this molecule, right? If you're a chem major or a biology major, if you're a biology major, I don't know if Dr. Barlow makes you learn molecules, but you don't need it for me, okay? You can see, though, where the ATP, where the name comes from here, because it's made up of an adenine, a ribose, and then there are 3 triphosphate, 1, 2, 3 inorganic phosphates that are attached to the adenosine. All right? So, one of the other energy molecules that's available in the muscle cell is ADP, which is adenosine diphosphate. So if I've got the diphosphate version and some inorganic phosphate, I can add them together and make adenosine triphosphate. Okay. So let me see if I can draw. I'm not very good on this whiteboard online, but I'll give it a go, see if I can draw you a picture here. There's my adenine and my ribose. It's too far away. I need to be able to make them smaller. Capital P. It's so much easier on the regular whiteboard. Okay, so this is our ATP. Right, we've got adenosine, and then we have energy bond, and an energy bond, and there's our 3 phosphates. That's going to turn into, we're going to break that down into, and the arrow can go both ways because we can build it back up again. So this time I'm not going to write out the whole word. We're going to have, let's do it this way, A, P, I, P, I, P, time we've got an energy bond, an energy bond, and we've released the energy bond here. So now we have adenosine diphosphate, an inorganic phosphate that gets released, and that energy bond is released and we can use it for muscle contraction. Okay? We also get a hydrogen ion released in this example here, which can cause problems because hydrogen ions are acidic. And we mentioned a little while ago, enzymes don't like it when the environment starts to turn to acidic. Okay? So, if we have energy available, we can rebuild our ATP in the opposite direction. So when we're making energy, we don't just make it for muscle contraction, we make it to do a load of the other jobs as well. Is that coming up for you? Yes, good. All right. Time's in. 1/2 past. Good. Okay. So when we break down the ADP, we get an inorganic phosphate, we get an energy bond that's released that we can use for muscle contraction or we can use for something else. And we get a hydrogen release, which we have to keep an eye on because if the hydrogens build up, then the enzymes just say, I quit, I'm not doing my job anymore. Okay? So, what we want to know then is, during my particular event, or at any point when I'm training, which energy system am I using, and which energy substrate am I using? Okay? So this depends a lot on how long the physical activity or exercise is going on for. It also depends upon how hard am I working, which is the intensity that I'm working at. Okay? So those two variables come together to really dictate which food source is being used, which energy system is producing ATP. Okay? If what I'm doing is relatively quick and high intensity, so I'm doing a 100 or 200 meter sprint, I'm throwing a shot or a javelin, I'm doing long jump or high jump, right? Then I'm going to do work that requires energy very quickly, and so we're going to use the anaerobic metabolic systems, right? So I don't need oxygen. If, though, I'm going to do something that is a little bit less hard work, and it's going on for a longer amount of time, anything longer than 3 minutes, now I've had time to get enough oxygen into my body that I can switch and use aerobic metabolism. Right? So the time and the level of work are the two key variables here. Okay? Now, we're talking about anaerobic systems in this chapter. So our two anaerobic systems are the ATP-PC system, which is our adenosine triphosphate, and the PC stands for phosphocreatine. And that energy system provides ATP for somewhere up to 25 to 30 seconds, depending which textbook you're looking at, right? And then the second energy system is called glycolysis, and that maybe rings some bells in your head because that word looks very much like glycogen okay? Possibly a little bit like glucose. So in glycolysis, we can use carbohydrate, and that will provide energy, ATP, from around 30 seconds up to about 2.5 minutes, possibly 3 minutes. Okay? So the ATP-PC system occurs in the sarcoplasm of the muscle cell, right? So in the kind of jelly substance of the muscle cell. And it does not use any type of food substrate directly, right? So it relies solely on energy bonds that are already available within the muscle cell. Okay? That's why it can't go for very long. Right? So we use the ATP-PC system for activities that need a lot of energy, or we also use it when we are changing intensity very rapidly. So if you're sitting in the classroom and the fire alarm goes off and you stand up and walk out of the classroom, that switch from being sedentary to standing up and moving would use the ATP-PC system. 100-meter sprint would use ATP-PC system because it takes how long? Right? 8-9 seconds. If you are doing a 1RM, you would use the ATP-PC system. Okay? So it can provide ATP for muscle contraction in a very short amount of time, but it can't last for very long at all. Okay? And that's because there's only a limited amount of ATP available just sitting in the muscle cell without being made, right? So without the energy systems doing anything, when you're just sitting there, there's some ATP floating around in the muscle cell. There's enough for about 5 seconds or so, okay? And then in 5 seconds, we still haven't managed to break down any glucose or glycogen, even though they're small molecules, you can't break them down in 5 seconds. So then we rely on phosphocreatine, which is another energy molecule in the muscle cell, but we can't use the energy from that molecule. So we steal the energy from the phosphocreatine and we rebuild the ATP. So, it's so much easier to do this on the whiteboard. Let's see. Here's my ATP. I'm going to have ADP plus PI plus hydrogen plus energy. Oops. Right. So here we've got ATP gets broken down into ADP, inorganic phosphate, hydrogen, and we release the energy. And that energy goes sailing off and gets used for muscle contraction. Okay? Now, we have a friend here helping us break down ATP. And our friend is an enzyme very easily called ATPase. So it's difficult to forget what ATPase does, right? ATPase breaks down facilitates breaking down ATP. Right? So this is good. We get like 5 seconds of this, but 5 seconds doesn't get us out of the building if the fire alarm is going off. Okay? So we don't want to just stop moving after 5 seconds, so we have to have a backup plan. All right? Our backup plan is phosphocreatine. Right? So we have phosphocreatine, and it gets broken down into creatine plus inorganic phosphate plus energy. Okay? And we have another little helper to facilitate breaking down phosphocreatine is called creatine kinase. Okay. So, here's how this works, right? We've got 5 seconds of ATP. And we break it down, we release the energy to get up out of our chair and walk towards the door of the classroom. ATPase facilitates that breakdown. Then I'm left with ADP and inorganic phosphate floating around. Right? So, what I can do is I can steal this energy, feed it back here, and rebuild ATP. Right? And I have maybe 20, so I've got 5 seconds worth of ATP. If I train this system really hard, I might get up to maybe 10 seconds worth of ATP. And I've got somewhere around 15 to 20 seconds worth of phosphocreatine. So this system, the ATP-PC system, can last for 25 to 30 seconds. Does that make sense? Now, if we want to rebuild phosphocreatine, so say you're a sprinter and your training session is not, sadly, only one sprint and you're done and you go home for the day, right? Your coach is going to make you sprint and sprint and sprint, all right? So how do I rebuild the phosphocreatine for the next lot of sprinting? So then I can do this. I can steal, when I break down my ATP, when I'm during my, what would you call it? During your rest phase, right? So whether your rest phase is walking or whether your rest phase is stationary, you're still going to make some energy. So now I can take that energy and I can add it in back here and rebuild phosphocreatine. But this is a rest phase or a low intensity phase. Questions? So it's very tempting to think that if I'm a sprinter or a thrower, that the fatigue I feel is due to the fact that I run out of ATP and phosphocreatine in the muscle cell. All right, I deplete that stored energy source. Okay. Now, the research doesn't seem to support that idea. We haven't talked about muscle fiber types yet, but when we do, a type 2 muscle fiber is the one that is most likely to use this energy system. And it would have enough energy at maximum contraction, which never happens in real life, right? You can stimulate it in the lab, but it doesn't happen in real life. It's got enough ATP and it's got enough phosphocreatine for another 4 seconds, right? So the timelines are different between the depletion of ATP and PC and a decrease in muscular force, which represents physiological fatigue. Okay? So we come back to our friend stroke enemy, the hydrogen ion. All right? It's thought at this point in time that the increase in acidity within the cell from the release of hydrogen ions by breaking down ATP is more likely to be the source of fatigue, of the drop in muscle force, right? Because that acidity impacts the enzymes, and the creatine kinase and the ATPase then slow down, and then we see force decrease, fatigue. Okay. If we do see close to or complete depletion of phosphocreatine, then as I showed on that diagram, there has to be an aerobic recovery period to rebuild phosphocreatine for the next short, high-intensity type work. Right? So if you're doing interval type training, okay, that's what the little rest phase is for. So you might sprint 50 meters, walk 25 meters, sprint 50 meters, or whatever, right? I don't do intervals, you can tell, right? A lot of anaerobic sports require that kind of downtime in them, okay? And at this point, go, creatine supplements, right? So I take creatine supplements, and then I'll have more phosphocreatine available, and I'll be able to go for longer than 25 or 30 seconds before we see muscular fatigue. Okay? There's, it won't, let me just check now, because I don't know if that's the right page number in this book. No, let me find that for you. Aha, it's on page 38 now, and it's now box 2.3, not box 2.8. Okay? So there's some information there about creatine supplements. So there are some pros and cons. Okay? The downside to creatine supplementation is that it appears that most people, a trade-off is water retention, right? That water retention can be 3 to 6 pounds. That's a lot of extra weight if you're a sprinter. Now, if you're a thrower, maybe 3 or 4 pounds of water isn't going to impact your performance in the same way. But if you're a sprinter, I don't think you can afford to have anywhere from 3 to 6 extra pounds of water weight. Right? That would slow you down. So that's a bit of an issue there. So far, it doesn't appear, although the athletic trainers would disagree with this, as far as I'm aware, there's not a lot of data on problems with creatine supplementation. The one thing that keeps coming up is kidney issues, so kidney stones or some kind of dysfunction in the kidneys. But there's not a lot of data on that yet. So we'll see where that goes. All right. The other thing about creatine supplements is that not very much of the supplement actually makes it into the muscle tissue. Right? And not all the creatine that makes it into the muscle tissue then becomes phosphocreatine. So you end up paying money for a supplement that may or may not actually improve performance. The average performance increase is less than 10% improvement. So it's not a big improvement. On the other hand, if you're a 100-meter sprinter and first and second is by 0.02 of a second, then less than 10% increase in performance might be helpful, right? So if you can find a way of taking the creatine without the water load, then maybe it's worthwhile, okay? So these kinds of ideas are useful for these short-term type events. For intermittent events like basketball, right? We know that the capacity of this system does improve with training, okay? And the capacity for recovery in the system improves with training. So there's lots of bonuses, right? For basketball players, soccer players, right? People who have to sprint and then get to maybe jog for a little while and then sprint again, right? Okay, we're going to stop there for today. Tomorrow, Monday, we'll look at what are the adaptations that can occur in this system, and then we'll look at the second anaerobic system. All right? So, if you didn't have a good look at Chapter 2 yet, make sure you have a decent look at it over the weekend so that maybe on Monday you'll have some other questions. I will try to look at it over the weekend as well and update my notes. And I will see you on Monday. Have a great weekend. Okay?